Thanks for the kind introduction, and thanks so much for the organizers for the invitation to speak this year. This is my first time at this conference, and I just want to say generally that it's such a fantastic group of folks that are very mission-driven, and I've found being involved in this community has been very fulfilling. I'm very excited to tell you today about our work with Oisin Biotechnologies and talking about some technology we've developed for a very specific and important task. So I don't really need to tell this audience about that aging sucks, and that eliminating age-associated disease is going to have a significant impact on humanity, and certainly the individual experiences of humanity. Obviously, aging is the most significant risk factor for many of the diseases that take us too early, and being able to address these in mass, is a critical challenge of our time. Also, many of you may know, and we believe very strongly, that one of the chief causes of aging are senescent cells. And there's a considerable amount of research and evidence and compelling studies to support this. Because Judy wasn't here to give a background on senescent cells, I'll just give a quick primer. I mean, senescent cells are cells in the body that, due to genotoxic, cytotoxic, other stresses, basically go into replicative arrest, and but don't die. So something has failed in their ability to go through apoptosis or to be cleared, and they'll continue to be metabolically active and continue to use energy and secrete factors. Now, this causes buildup of active enzymes in their lysosomes, usually characterized by an accumulation of beta-galactosidase, which can be stained using XGAL and makes them obvious in many cases. But the most important thing, health-wise, I would say, is that these metabolically active cells are giving off a lot of things that are deleterious to the surrounding area, and I would argue systemically. So the senescence-associated secretory phenotype has been well characterized, results in the secretion of many signaling cytokines that circulate throughout the body, some local metalloproteinases that wreak havoc in the local microenvironment, and really have been demonstrated in many very important and high-impact studies over the past 5 years to be associated with pretty much every single age-related degenerative disease that we're aware of. And so it's really, this foundational science is really pushed, fortunately, a lot of activity in developing compounds called Senolytics that can selectively clear these senescent cells, kill the senescent cells, or other approaches. And so that's the task that we've decided to focus our efforts on. And I'll just highlight a couple of things that are really important to our approach. So we know from the fantastic studies of the Mayo Clinic that if you generate a genetically modified mouse where you're able to selectively ablate senescent cells over its lifetime, so the littermates after a while, as they get older, show significant differences. So we see a 25% median lifespan increase. We see significantly less cancer. These two mice here, the same age, same genetics, same diet, same lifestyle, and they have significantly less cataracts, frailty. There's less loss of hair, significant differences. Now, it's important to note that these are genetically modified mice, and they've been treated, since birth to eliminate their senescent cells. So, fortunately, there's also some great data out there suggesting or supporting the fact that you can intervene late in life. So once a lot of these conditions have already progressed to a point where they're obvious, and this is a great study by Peter de Keyser's group, where he was able to ablate senescent cells both in two models, one in a fast-aging TTD model and in naturally aged mice using a P53-based approach, and saw significant improvements in a lot of these age-related phenotypes, including fitness, fur density, and renal function. And also in that study, and others have shown that you can ameliorate, toxic damage from other sources such as chemotherapy. So we have ample evidence now that not only can we, that senescence is principally involved in the process of aging, but also that we have an opportunity through the ablation of senescent cells to ameliorate and even reverse some of these phenotypes. So at Oisin, we decided to utilize, so based on this clear data, I mean, we can argue about what makes a senescent cell, whether it's P16 or multifactorial approaches. Well, we can all agree that in these mice, when they ablate a certain cell based on its genetics, they see a substantial improvement. And so that's what we wanted to replicate in some sort of systemically administered therapy. So we wanted to utilize a strategy that had already been shown to work in the models that we have. Now, because we're going to be administering these therapies to relatively healthy people, so it needs to be something that can be systemically delivered, is extremely well tolerated at therapeutic doses in healthy and aged individuals. And it also needs to be suitable for repeated or prolonged dosing. It also, because these diseases, happen in almost every tissue of the body, you need broad bioavailability. And finally, because senescent cells, for instance, do not represent a gigantic burden of cells in your body. Estimates range from 1 to 5% and up to 30% maybe in certain tissues. But ideally, a therapy has to have broad bioavailability, but be highly selective for those damaged cells while sparing the healthy cells around it and even spurring on repair. So Oisin has decided to adopt a genetic approach that we feel is extremely selective. And I'd like to show this slide because it really compares it to how far we've come as far as drugs go. So surgery has been really, I have a lot of experience in the cancer field. Surgery has worked really well. If you can see a cancer, you can cut it out, and many times it's a cure. But it's really an indiscriminate attack on the cells you're looking at. And with senescent cells, it's just not an option. Chemotherapies and a lot of the small molecule drugs, attack something very broad in a cell, like metabolism. It's kind of an important thing. And so as a result, you may achieve the goal that you want, but you have significant off-target effects. And now we're in an age where we have, we're increasing our selectivity through specific antibodies, antibody-drug conjugates, and CAR-T approaches. And these are targeting cells based on their surface markers. And as our CEO, Matt Scholes, likes to say, it's like targeting every redhead in the room. So at least you can identify the redheads and kill them, but it's not necessarily something you want to do. So so we thought we would want to adopt an approach that had shown success in the past, and that's really targeting cells based on what they're thinking, or in essence, what transcriptional pathways are activated. And so to summarize our platform, we're using a lipid nanoparticle approach. It's a novel type of nanoparticle that I'll be describing, carrying a plasmid-based gene therapy that provides extreme selectivity for senescent cells. So in general, we have what's called a Fusogenix lipid nanoparticle platform. So this is a well-tolerated, broadly applicable gene therapy platform. And we have a DNA plasmid that really confers selectivity by using selective engineered promoters to drive suicide genes and eventually lots of other things as well. And we can engineer these promoters to be selective for certain types of cells, avoid other certain types of cells by adding elements like enhancers and repressors to really make them exquisitely specific for the cells we're targeting. And altogether, we've created several versions of this that can be either induced to kill cells or are self-inducing, and I'll explain that in a second. So the most important part is the DNA payload. This is a highly selective suicide gene in that, as I mentioned, we have a selective promoter. So sort of the canonical promoter that we're using to target senescent cells is P16. We're using an engineered specific version of a P16 promoter that generally targets senescent cells. And we also have other versions as well. So we have a P53 targeting construct that we're using for some oncology studies and for aging studies. And I guess the point here is it's like writing code and injecting it into a cell. So we can write a code in a DNA strand and selectively deliver it to cells and run that code in the cell and really hijack the machinery of the cell to accomplish the task that we want. In this case, we're mainly driving an inducible version of caspase-9. So it drives the cell into apoptosis. Caspase-9 for us is a very good way to induce apoptosis in cells because it's based on the endogenous caspase-9. It functions really late in the apoptotic pathway, so there's not really much chance for the cell to escape. It'll bypass any upstream abnormalities such as BCL2 overexpression and other abnormalities. And its induction of apoptosis, which is really important for senescence, is independent of the cell cycle. So the way we do it is through an inducible version of caspase-9. So we use a version where its dimerization domain has been, its CARD domain has been cleaved off. We've got a dimerization domain based on FKBP inserted in there, and then we have different versions of the dimerization domain that perform different functions. So we built 3 main versions. The first of those is very similar to those used in the initial studies by the Mayo Group, using a dimerizer called AP1903 is the clinical version of that. We also have another version that's dimerization is induced by rapamycin, which this group will be familiar with. We've also created versions that will auto-dimerize, self-dimerize at different rates, allowing us to sort of titrate in the suicide gene and induce death. So once you induce the dimerizer, these 2 caspase-9 constructs will come together. They'll basically autocleave each other and be recruited to the apoptosome and induce cell death in those cells. So that's the idea. So does it actually work? So number one, it's a multi-component system. So we have a plasmid gene therapy and an LNP. We're delivering that first. We're letting that express in the cells that we want to express it in. And then we're giving a small molecule dimerizer in the case of those first 2. So first of all, we want to make sure that adding the dimerizer itself is not a limiting factor. So this is looking at the IC50 for AP1903. We find that we have low single-digit picomolar IC50 for this molecule. It's extremely potent at very, very low concentrations. And we know there's clinical phase 2 experience with this molecule. It's got very broad bioavailability. So I think we're going to be able to achieve therapeutic levels of this drug in vivo very easily. There's a lot known about rapamycin. In our rapamycin constructs, we're also getting very low picomolar IC50s for dimerization of our caspase-9 constructs. It's not quite as strong as the AP1903, but well within any therapeutic window that we want to use in vivo. So we're very optimistic, enthusiastic about using rapamycin to dimerize these constructs. And I won't spend too much time, but we have also a self-dimerizing construct. So one of the things that I guess one of the limitations of a dimerizer is the fact that you're basically killing all of the cells within a couple hours. So we've introduced self-activating auto-dimerizing system in order to sort of slow down that killing. It really depends. If you're a smoker and you have a lot of senescent cells, you might want to slow that down a bit. If you're a relatively healthy individual, it might not matter too much. So in an experimental system, we can induce the expression of P16 by irradiating cells with 10 grays of radiation. So I'm showing here that we can induce the production of P16 protein. If we use our construct with a P16 engineered promoter and drive luciferase, we see that as we produce P16 protein, we're also inducing the production of luciferase. So that agrees well. In this case, we're looking at the induction of our inducible caspase-9. So I'm showing you a Western blot. Anyway, so you can see the band. I wonder if I got the mouse. Yeah, so basically caspase-9 is what the big bands across the gel. In a cell where we've treated them with 20 grays of radiation without dimerizer, we see the induction of an ICAS9 specific band. It runs a little bit different than caspase-9. And then when we add the dimerizer, this band disappears, indicating those cells are disappearing. And then when we look at the viability of these induced senescent cells, with the addition of the dimerizer, we see a significant reduction in their viability, indicating or suggesting that we're killing these cells through apoptosis. Then we can use sort of a sophisticated imaging flow cytometry method to look at, test the fact whether they kill by apoptosis. So this basically looks at the nuclear morphology and has an algorithm to calculate whether they're going through apoptosis, and we see we're getting a dramatic induction of apoptosis in these cells that have had induced senescence, treated with the plasmid treatment, and then induced with dimerizer. So now we've got a plasmid-based selective system to selectively kill senescent cells. We did a few neat imaging experiments to really demonstrate this because, like I said, the selectivity is important. So what I'm going to show you here are in vitro cells growing in culture. On the left side, the green cells, all the green cells are highly p16 positive, and we're taking a movie, basically taking a shot every 5 minutes from the time we add dimerizer, and you can see these cells are pretty much obliterated within 6 or 7 hours, and the majority of the activity happens within the first 2 hours. What's really important, though, is in a background of non-p16 positive cells, if we have sort of 4 or 5% p16 positive cells on the right here, when we add dimerizer here, we can see we're selectively driving those cells to go through apoptosis and killing them. So we have a selective, highly selective method to ablate p16 cells. So that's great. So we have a plasmid. Now I want to talk to you about how can we make this into a drug that's actually going to work in people. And so I don't know how many people are familiar with the gene therapy field, but in vivo gene delivery is really challenging. So there have been some ways to sort of solve different aspects of it. None of those have been optimal. Nucleic acid therapies really it's challenging to get to their targets, because to have a therapeutic effect, you need to get to the cell of interest, get the cargo inside the cell where it's going to have its activity. And even getting it to the sort of the organ in the area is really not enough. It has to penetrate the cell membrane and be intracellular. And so the cell membrane has obviously evolved over millions and millions of years to repel things, right? So it'll either repel things on the outside through the immune system, but if it takes things up, it'll typically merge them with lysosomes and break it down with acid. So most current drug delivery systems use really one of two methods. There are viral-based delivery systems, so AAV and lentivirus. And these have, I think for the list of requirements for a systemic senolytic really don't align well with viral-based approaches, because they can only be administered once. They have limited cargo carrying capacity. They're extremely expensive to make. So to make a systemic dose would be in the order of $1 million a dose. So it's really not feasible to go with a viral route. And so we looked initially at non-viral route. Lipid nanoparticles, well-established platform for in vivo drug delivery. They provide improved protection, improved pharmacodynamics, and you can tune them somewhat by adding groups on the outside. But all of the current clinical stage lipid nanoparticles use a similar platform. And you may be surprised to know that their efficacy is dependent upon a single biophysical property, and that's having positively charged or conditionally positively charged lipids. And positive charged lipids interact with membranes and basically poke holes in them. You can imagine if you're poking holes in membranes that you can potentially introduce toxicity. And the thing about nanoparticles, when they're administered systemically, the body has a very good filtration system to take them out of the bloodstream, and that's the liver and the spleen. So you're going to get, at least 50%, sometimes up to 90% of the injected dose is going to be taken up by the liver and spleen. So all of the currently commercialized and approved platforms have this issue with liver uptake and toxicity. So it's really been difficult to use LNPs and to be able to target tissues outside the liver. So, and I like to show this slide because it really hammers home the safety issues that LNPs have had due to this issue. So on the top, I'm showing these are clinical programs. The 3 top programs are using fully cationic, positively charged lipids, and I want to direct your attention to the maximum tolerated dose. So sub 1 mg per kg. Very, very low doses. All these programs failed in phase 1 or phase 2. A second generation of lipids were discovered over the past 10 years. The first approved product by Alnylam last year is using one of these lipids. They're better tolerated, so in the 1 mg per kg range, but again, not well tolerated enough to target anywhere outside the liver. You'll notice that Alnylam's approved program is for a liver-based disease, and no platforms using lipid nanoparticles have been able to target programs outside the liver successfully to date. So we adopted a different approach. We didn't want to use toxic cationic lipids, but we do know that viruses are useful for injecting DNA in cells, and one of the reasons why they're so useful is that they've evolved very elegant fusion mechanisms to be able to bind to the outside of cells and inject their cargo inside. So we took a look at some of the fusion proteins from different viruses. This is just a couple examples. In general, they're really cool. They're very elegant. They can attach to membranes, bring them together, mix the lipids, and create a pore. But as you can see here, they're quite large. They have large ectodomains, which means they're immunogenic, and they're obviously very, very difficult to manufacture. So fortunately, about 13 years ago, Canadian scientist Roy Duncan at Dalhousie University discovered this new class of fusogenic orthoreoviruses that have a very cool phenotype. When they infect the cells, and they infect things like alligators and birds, they use a fusion protein to cause all of the cells around the infected cell to fuse together. And so he spent a number of years trying to isolate the protein component that caused this effect, and found the smallest known fusogenic protein by 2 orders of magnitude. So this is a very, very tiny single transmembrane domain protein that is sufficient to induce cell-to-cell fusion. And he went on and spent much of his career characterizing a number of these different fusion proteins from different versions of the fusogenic orthoreovirus. And so we spent a number of years in my academic lab taking the best, most active parts of these fusogenic proteins to create an ideal, highly active fusogenic protein for drug delivery. And when we formulate those fusogenic proteins into a lipid nanoparticle that does not contain positively charged lipids, we get really effective delivery into cells. And I like to show this slide because if you're putting DNA with lipids in a dish on cells, every cell gets roughly the same dose. But if you inject that same formulation in a human, you get dramatically different doses to different tissues. So your liver will get 75% of the dose, and your other cells will get 1 to 2% of the dose. And so what we've done here is we're showing the difference between concentration and toxicity, concentration and efficacy of a cationic lipid formulation compared to the formulation we're using with fusogenic proteins. And you can see certainly the toxicity is low at low concentrations with both platforms. As you increase the concentration, our formulation is non-toxic all the way up to massive doses, while cationic lipids will kill every cell. And again, with efficacy, we can raise the dose considerably and achieve maximal efficacy without toxicity. And this is really important for systemically administered therapy. Okay, so now we've got a really a neat selective DNA payload we can use to selectively kill cells. We've got a lipid nanoparticle platform we believe is going to be well tolerated and effective for this gene delivery. So our initial studies, we just asked, can we use this platform to selectively ablate senescent cells in aged mice? So we took, in this first study, 16 aged mice, 80 weeks of age, so getting close to 2 years. We gave them 2 doses of a lipid nanoparticle with a P16 promoter-driven suicide gene, and each animal received either a single low or high dose. And so first we asked the question, in different tissues, can we see a reduction in P16 expressing cells? And we saw in almost every organ we looked at a dose-dependent reduction in P16 expressing cells as measured by RT-PCR. Back when we started this, we weren't awesomely good at staining for beta-galactosidase, so I'm showing you the best images. But we saw in all the tissues we looked at, we saw a great trend of reduction of beta-galactosidase positive staining cells in many of the organs. And so we were comfortable that this combination of P16 suicide gene-based payload with an LNP platform was going to be able to selectively ablate senescent cells. So we then started a lifespan study in 41 aged mice. So these guys were older, just over 2 years of age at 106 weeks. And in this case, we decided we divided them basically evenly into control group that just received a non-payload formulation, a formulation that contained our P16-driven ICAS9, a formulation with the P53-driven ICAS9, and then a combination of the 2 that received sort of half of dose of each. And they were dosed once a month on centers until they died. And actually, I have to update the slide. So this study is now over, and I'm happy to share with you some of the results of what we saw. So first of all, I'm going to just show you a snippet of data. In vivo, we actually adapted our image stream imaging flow cytometry assay to identify senescent cells using XGAL staining, or sort of beta-galactosidase staining with XGAL. And so we saw a really nice reduction of senescent cell burden in a number of tissues. And we were actually kind of surprised to see that the P16 sometimes had a very marked reduction. P53 didn't actually do a bad job either. What was nice, and in the combination at the half dose, we saw really, really, really tight results and a strongly significant reduction in these XGAL positive beta-galactosidase positive cells. Because these mice were frail, we didn't want to do a lot of, we were thinking about doing activity assays and a number of other things, so we did do their bone density, and we saw a significant increase in the bone density of those, of just the male mice. We had a few more male mice in the cohort. Significant increase in bone density in the mice receiving the combo treatment. It's really, this is I would say by longevity standards, this is a smaller study, but I'll just show you the median lifespan results. So in the control group, median lifespan was 119 weeks, which tracks really well with other studies such as Charles River cohort studies. If they received just the P53-driven ablative therapy, extended their median lifespan to 133 weeks, or an increase of about 11.8%. If they received just the P16 ablative treatment, increase in median lifespan of almost 18%, and you can see really shifting that curve. And what was really exciting is that with the combo treatment, we actually didn't have a single mouse die until halfway through the experiment. The control mice had already achieved their median lifespan, and at the end of the study, we had 30% of the mice alive. And I'll say, I actually did the study in a university academic atmosphere that's used to oncology studies, and they really didn't know how to handle mice that were getting old, because they would have, some of them get lesions, some of them would look frail, and they say, oh, this guy looks frail, should we kill him? I'm like, no, that's not the point of the study. We want to keep them alive as long as possible. So we have to educate them a little bit more on these kind of studies. So I just want to spend the last minute talking about, so we have, I think, a very compelling platform to bring to the clinic. Obviously, when you're thinking about systemic delivery of drugs in a human, you need to think about a lot of things other than does it work. For one thing, is it immunogenic? So because we're using a viral protein, does it produce antibodies that will either neutralize it or create a hypersensitivity response? I'm happy to say that we've done a ton of these studies. We don't get any reactive antibodies produced. We don't get any hypersensitivity. We've done now 2 non-human primate tox studies, well tolerated at more than 10 times the estimated human dose. And we don't see, and most importantly, we don't see any histological changes in the liver or spleen. We've also looked at biodistribution in non-human primates, and we do get good lung and liver uptake, but actually we get a good therapeutic dose to many other tissues, including there's a relatively good dose to the brain and other tissues that we'll be digging deeper in this current study to see what specific cell types we're hitting. I'll skip over this just to say that our goal now is to ramp up production. So we're using a scalable micropolitic manufacturing platform to create these for GMP production that'll be online in July 2019. And this theme's come up again and again in this conference. We're not going to go to clinic with an age-related disease indication, but we have found, and I didn't have time to go through this today, that the P53 version of this therapy works exceptionally well in solid tumors. So Oisin has spun out an oncology therapeutics focused company, Fusogenix. We just raised our pre-seed round. It's based on P53 transcriptional targeting of solid tumors, and we've actually designed and are getting ready to launch a phase 1 clinical trial starting in early 2020. Fortunately, so I guess the kind of the people we'll be treating in that study, there'll be a lot of elderly people with advanced stage disease. So we'll use that opportunity to collect information on age and senescence-related biomarkers. I'll leave it there because I went over time. I apologize for that, but we have a great team at Oisin: Gary Hudson, Matt Scholes, Eric Garcia, Steve Hilbert, and our many advisors, and a fantastic team that actually did this work. And I appreciate your attention.